Chapter 10: Analyzing Genes and Genomes. The 21st century promises to be an exciting time for cell biology. New methods for analyzing and manipulating DNA, RNA, and proteins are fueling an information explosion and allowing us to study genes and cells in previously unimagined ways. We now have access to the sequences of many billions of nucleotides, providing the molecular blueprints for dozens of organisms, from microbes and plants to birds, insects, humans, and other mammals. And powerful new techniques are helping us to decipher this information, allowing us not only to compile huge, detailed catalogs of genes and proteins, but to begin to unravel how these components work together to form functional cells, tissues, and organisms. The goal is nothing short of obtaining a complete understanding of the process that takes place moment to moment inside all living things. This technological revolution has been powered, in large part, by the development of methods that have dramatically increased our ability to handle DNA. In the early 1970s, it became possible for the first time to isolate a given piece of DNA out of the many millions of nucleotide pairs in a typical chromosome. This in turn made it possible to generate new DNA molecules in the test tube and to introduce this custom-made genetic material back into living organisms. These developments, dubbed Recombinant Dna Technology or genetic engineering, make it possible to create chromosomes with combinations of genes that could never have formed naturally, or combinations that could conceivably occur in nature, but might take thousands of years of chance events to come together. Of course, humans have been experimenting with DNA, albeit without realizing it, for millennia. Modern garden rose varieties, for example, are the product of centuries of selective breeding between strains of wild roses from China and Europe. Figure 10.1a. Similarly, the enormous variation in the sizes, colors, shapes, and even behaviors of different breeds of dogs is the result of deliberate genetic experiments, with selection for desired traits, carried out since the gray wolf, the ancestor of the modern dog, was first domesticated some 10,000 to 15,000 years ago. Figure 10.1b. Modern genetic engineering techniques, however, allow us to alter DNA with much greater precision and speed with the latest equipment and techniques. Even a beginning student can now collect an organism's genetic material, isolate a region of DNA containing a specific gene, produce a virtually unlimited number of exact copies of this DNA, and determine its nucleotide sequence with relative ease. Using variations of these engineering techniques, the isolated gene can be redesigned in the laboratory and then transferred back into cells in culture to elucidate its function. With slightly more sophisticated methods, the engineered genes can be inserted into animals and plants, so that they become a functional and heritable part of the organism's genome. These technical breakthroughs have had a dramatic impact on all aspects of cell biology. They have made possible our present knowledge of the organization and evolutionary history of the complex genomes of eukaryotes, as discussed in Chapter 9, and have led to the discovery of whole new classes of genes, RNAs, and proteins. They provide new ways of determining the functions of proteins and of individual domains within proteins, revealing a host of unexpected relationships among them, and they give biologists an important set of tools for unraveling the mechanisms by which a whole animal or plant can develop from a single cell. Recombinant DNA technology has also had a profound influence on many aspects of human life outside scientific research. It is used to detect the mutations in DNA that are responsible for inherited disorders and to diagnose an individual's predisposition to genetic diseases such as cancer. It is used in forensic science to identify or acquit suspects in a crime. It is used to produce an increasing number of human pharmaceuticals, including insulin for diabetics and the blood clotting protein factor VIII for hemophiliacs. Even our laundry detergents, which contain heat-stable proteases that digest food spills and blood spots, make use of the product of DNA technology. Of all the discoveries described in this book, those that led to the development of recombinant DNA technology are likely to have the greatest impact on our everyday lives. In this chapter, we describe the most common methods for manipulating genes and determining their function. We first survey the principal methods of the revolutionary field of recombinant DNA technology, beginning with a discussion of the basic techniques of DNA analysis. Next, we describe how DNA sequences can be isolated and produced in large numbers by the techniques of DNA cloning and the polymerase chain reaction, PCR, and we explain how these sequences can be used to produce and study proteins. In the final section of the chapter, we examine how DNA technology is used to discover the roles of individual genes and proteins in cells and in organisms. Together, these techniques have made possible our understanding of biology, including much of the material presented between the covers of this book. Manipulating and analyzing DNA molecules. Until the development of recombinant DNA techniques, crucial clues for understanding how cells worked remained locked in the genome. Once scientists realized that genetic information was encoded in the sequence of nucleotides in DNA, they needed to get their hands on individual genes to see what they look like and determine how they function. Before the revolution in DNA technology that took place in the 1970s, this task was almost impossible. Part of the problem was that isolating a single gene was not a trivial matter. Unlike a protein, a gene does not exist as a discrete entity in cells, but rather as a part of a much larger DNA molecule. Even bacterial genomes, which are much less complex than the chromosomes of eukaryotes, are enormously long. The E. coli genome, for example, contains 4.6 million nucleotides. Large pieces of DNA can be broken into small pieces by mechanical shear. However, the fragment containing a particular gene will still be only one among 100,000 or more DNA fragments that would be obtained, for example, from a mammalian genome by these means. And in a sample containing many identical copies of the same large DNA molecule, each molecule would be broken up differently by shear, producing a confusing set of random fragments. How then can a gene be isolated and identified? The solution to this problem emerged in large part with the discovery of a class of bacterial enzymes known as restriction nucleases. A nuclease catalyzes the hydrolysis of a phosphodiester bond in a nucleic acid. But these enzymes have a property that is distinct from other nucleases. They cut double-stranded DNA only at particular sites, determined by a short sequence of nucleotide pairs. Restriction nucleases can therefore be used to produce a reproducible set of specific DNA fragments from any genome. We begin this section by describing how these enzymes work and how the DNA fragments produced by them can be separated from each other. We then discuss how these fragments can be probed to identify the ones that contain the DNA of interest. Restriction nucleases cut DNA molecules at specific sites. Like most of the tools of DNA technology, restriction nucleases were discovered by researchers studying a specialized biological problem that had gripped their interest. Certain bacteria, it was noticed, always degraded foreign DNA that was introduced into them experimentally. A search for the cause of this targeted destruction revealed a novel class of nucleases present inside the host bacterium that cleaves DNA only at certain nucleotide sequences. The bacterium's own DNA is protected from cleavage by chemical modification of these same sequences. Because these enzymes restricted the transfer of DNA between certain strains of bacteria, the name restriction nuclease was given to them. This solution to a seemingly arcane problem in bacterial biology forever changed the way in which cell and molecular biologists study life. Different bacterial species contain different restriction nucleases, each cutting at a different specific sequence of nucleotides. Figure 10.2. Because these target sequences are short, generally 4 to 8 nucleotide pairs, sites of cleavage will occur purely by chance in any long DNA molecule. Thus, restriction nucleases can be used on DNA from any source. The main reason they are so useful is that a given enzyme will always cut a given DNA molecule at the same sites. Thus, for a sample of DNA from a human, treatment with a given restriction nuclease will always produce the same set of DNA fragments. Restriction nucleases are now sold by many companies and are typically ordered through the mail. One online catalog lists hundreds of such enzymes, each able to cut a different DNA sequence. The target sequences of restriction nucleases vary in the frequency with which they will occur in DNA. As shown in Figure 10.2, the enzyme He3 cuts a particular sequence of 4 nucleotide pairs. This sequence would be expected to occur purely by chance approximately once every 256 nucleotide pairs, 1 in 4 to the 4. By similar reasoning, the enzyme NOT1, which has a target sequence that is 8 nucleotides long, would be expected to cleave DNA on average once every 65,536 nucleotide pairs, 1 in 4 to the 8. The average sizes of the DNA fragments produced by different restriction nucleases can thus be very different. This feature makes it possible to cleave a long DNA molecule into the fragment sizes that are most suitable for a given application. Gel electrophoresis separates DNA fragments of different sizes. After a large DNA molecule is cleaved into smaller pieces with a restriction nuclease, it is often desirable to separate the DNA fragments from one another. This is usually accomplished using gel electrophoresis, which separates the fragments on the basis of their length. The mixture of DNA fragments is loaded at one end of a slab of agarose or polyacrylamide gel, which contains a microscopic network of pores. A voltage is then applied across the gel slab. Because DNA is negatively charged, the fragments migrate toward the positive electrode. The larger fragments migrate more slowly because their progress is more impeded by the agarose matrix. Over several hours, the DNA fragments become spread out across the gel according to size, forming a ladder of discrete bands, each composed of a collection of DNA molecules of identical length. Figure 10.3a. Physically isolating a particular DNA fragment from this agarose slab is fairly simple. A small section of the gel containing the band can be cut out using a scalpel or a razor blade, and the DNA can then be extracted. DNA bands on agarose or polyacrylamide gels are invisible unless the DNA is labeled or stained in some way. One sensitive method of staining DNA is to expose it to a dye that fluoresces under ultraviolet light when it is bound to DNA. Figure 10.3b. An even more sensitive detection method involves incorporating a radioisotope into the DNA molecules before electrophoresis. 32P is often used, as it can be incorporated into the phosphates of DNA and emits an energetic beta particle that is easily detected by the technique of autoradiography, figure 10.3c. Hybridization provides a sensitive way to detect specific nucleotide sequences. Exposing a gel to a fluorescent dye that binds to DNA or labeling the DNA with 32P will allow every band on the gel to be seen, but it does not reveal which of those bands contains a sought-after segment of DNA. To identify a fragment that has the nucleotide sequence of interest, one can take advantage of the fact that any single strand of DNA will form Watson-Crick base pairs with a second strand of complementary nucleotide sequence. Under normal conditions, the two strands of a DNA double helix are held together by hydrogen bonds, which can be broken by heating the DNA to around 90 degrees Celsius, or by subjecting it to extremes of pH. Such treatments release the two strands from each other but do not break the covalent bonds that link nucleotides together within each strand. If this process is slowly reversed, that is, by slowly lowering the temperature to normal body temperature, or by bringing the pH back to neutral, the complementary strands will readily reform double helices. This process is called hybridization or renaturation, and it results from a restoration of the complementary hydrogen bonds. Figure 10.4. A similar hybridization reaction will occur between any two single-stranded nucleic acid chains, DNA/DNA, RNA/RNA, or RNA/DNA, provided they have complementary nucleotide sequences. The fundamental capacity of a single-stranded nucleic acid molecule to form a double helix only with a molecule complementary to it provides a powerful technique to detect specific nucleotide sequences in both DNA and RNA. Hybridization is carried out using DNA probes designed to recognize a desired nucleotide sequence. To search for a nucleotide sequence by hybridization, however, one first needs a piece of nucleic acid with which to search. This DNA probe is a single-stranded DNA molecule, typically 10 to 1,000 nucleotides long, that is used in hybridization reactions to detect nucleic acid molecules containing a complementary sequence. Today, DNA probes of any desired sequence can be synthesized non-enzymatically. Machines the size of a microwave oven can be programmed to string nucleotides together by chemical synthesis to produce single-stranded DNA chains of any sequence up to several hundred nucleotides in length. Of course, to know what sequence to synthesize, one needs to know the nucleotide sequence of the DNA fragment of interest. Say, for example, one wishes to identify a DNA fragment that contains a portion of the beta-globin gene. Because the sequence of this gene is known, producing a probe that will recognize it is a straightforward exercise in looking up the sequence in a database and then programming the nucleic acid synthesizer. Nowadays, such probes are usually ordered from companies that synthesize custom-made pieces of DNA. Simply send the company an email containing the desired sequence, and the DNA molecules can be delivered the next day. Once the probe is in hand, it can be used to search for nucleic acids with a complementary sequence, including the fragment of interest among a collection of DNA fragments that have been separated by size on an agarose gel. The resulting hybridization with bands on an agarose gel can be visualized by southern blotting, a common laboratory procedure named after the scientist who invented it. Figure 10.5. DNA probes are widely used in cell biology. Later in the chapter, we will see how hybridization with specific probes can be used to determine in which tissues and at what stages of development a gene is transcribed. But first, we discuss how hybridization facilitates the process of DNA cloning. DNA cloning. We have seen that DNA molecules can be cleaved into shorter fragments by restriction nucleases, and that these fragments can be separated by gel electrophoresis. We have also seen that hybridization can be used to pick out a match with a DNA probe of known sequence. In this section of the chapter, we shall see how these procedures are combined to obtain a physical piece of DNA from a genome. In other words, we will discuss how a particular piece of any genome can be cloned. The term DNA cloning literally refers to the act of making many identical copies of a DNA molecule. It is this amplification that makes it possible to separate a particular stretch of DNA, often a gene of interest, physically from the rest of a cell's DNA, which is present in vanishingly small quantities compared with the final mass of the amplified sequence. Producing many copies of a defined segment of DNA from a genome is one of the most important feats of recombinant DNA technology, as it is a starting point for understanding the function of each stretch of DNA within the genome. DNA ligase joins DNA fragments together to produce a recombinant DNA molecule. Modern DNA technology depends both on the ability to break long DNA molecules into conveniently sized fragments and on the ability to join these fragments back together in new combinations. Any DNA molecule so constructed in the laboratory is called recombinant DNA. The cell itself has provided the tools with which to perform these molecular manipulations. As discussed in Chapter 6, the enzyme DNA ligase reseals the nicks in the DNA backbone that arise during DNA replication and DNA repair. See figures 616 and 626. This enzyme has become one of the most important tools of recombinant DNA technology, as it allows scientists to join together any two DNA fragments. Figure 10.6. Because DNA has the same chemical structure in all organisms, this simple maneuver allows DNAs from any source to be united. In this way, isolated DNA fragments can be recombined in the test tube to produce DNA molecules not found in nature. Once two DNA molecules have been joined by a ligase, the cell cannot tell that the two DNAs were originally separate and will treat the resulting DNA as a single molecule. If such a foreign stretch of DNA is appropriately introduced into the DNA of a host cell, it will be replicated and transcribed as if it were a normal part of the cell's own DNA. Recombinant DNA can be copied inside bacterial cells. Obtaining many identical copies of a defined stretch of DNA, often a gene, can be accomplished in several ways. One way is to introduce the DNA to be copied into a rapidly dividing bacterium. Each time the bacterium replicates its own DNA, it also copies the introduced DNA. DNA can be introduced into bacteria by a mechanism called transformation. Some bacteria naturally take up DNA molecules present in their surroundings by pulling the DNA through their cell membrane to the inside of the cell. The incoming DNA is often then incorporated into the genome by homologous recombination. The term transformation originated from early observations of this phenomenon, in which it appeared that one bacterial strain had become transformed into another. The deliberate transformation of one strain of bacterium with purified DNA derived from another strain provided one of the first experimental proofs that DNA is indeed the genetic material. See How We Know, pages 174 to 176. In a natural bacterial population, a source of DNA for transformation is provided by bacteria that have died and released their contents, including DNA, into the environment. In a test tube, however, bacteria such as E. coli can be coaxed to take up recombinant DNA that has been created in the laboratory. Figure 10.7. A great advantage to the experimenter is that naked DNA from any source, not just the DNA of the same bacterial species, can be introduced by this route. Bacterial transformation thus allows DNA from complex organisms, such as humans, to be studied easily in the laboratory. Specialized plasmid vectors are used to clone DNA. As mentioned previously, DNA introduced into bacteria under natural circumstances often becomes part of the bacterial genome. Investigators interested in cloning, however, find it easier to manipulate, copy, and purify their recombinant DNA when it is maintained as an independent molecule, separate from the bacterial chromosome. To maintain foreign DNA in a bacterial cell, a bacterial plasmid is used as a carrier or vector. The plasmids typically used for gene cloning are relatively small circular DNA molecules of several thousand nucleotide pairs that can replicate inside a bacterium. Figure 10.8. A plasmid vector contains a replication origin, which enables it to replicate in the bacterial cell independently of the bacterial chromosome. It also has cleavage sites for restriction nucleases so that the plasmid can be conveniently opened and a foreign DNA fragment can be inserted. Plasmids also usually contain a gene for some selectable property, such as antibiotic resistance, which enables bacteria that take up the recombinant DNA to be identified easily. Plasmids occur naturally in many bacteria. They were first recognized by physicians and scientists because they often carry genes that render their host bacteria resistant to one or more antibiotics. Indeed, historically potent antibiotics, penicillin for example, are no longer effective against many of today's bacterial infections because plasmids that confer resistance to this class of antibiotic have spread among bacterial species by horizontal transfer. See Figure 9.17. The plasmids used for recombinant DNA research are basically streamlined versions of these naturally occurring plasmids. To insert a piece of DNA into a plasmid, the purified plasmid DNA is exposed to a restriction nuclease that cleaves it in just one place, and the DNA fragment to be cloned is inserted into it by using DNA ligase. Figure 10.9. This recombinant DNA molecule is then introduced into a bacterium, usually E. coli by transformation, and the bacterium is allowed to grow in nutrient broth, where it doubles in number every 30 minutes. Each time it doubles, the number of copies of the recombinant DNA molecule also doubles, and after just a day, hundreds of millions of copies of the plasmid will have been produced. The bacteria are then lysed, and the plasmid DNA is purified, by virtue of its small size, from the rest of the cell contents, including the large bacterial chromosome. The purified preparation of plasmid DNA will contain millions of copies of the original DNA fragment. Figure 10.10. This DNA fragment can be recovered by cutting it cleanly out of the plasmid DNA with the appropriate restriction enzyme and separating it from the plasmid DNA by gel electrophoresis. See Figure 10.3. These steps effectively allow the purification of a given stretch of DNA from the genome of any organism. Genes can be isolated from a DNA library. We have seen how any DNA fragment can be produced in large numbers by inserting it into a bacterium. But how are these DNA fragments identified and chosen in the first place? More specifically, Without knowing each gene sequence in advance, how were individual human genes first isolated? As an example, we will describe the cloning of the gene for the human blood clotting protein factor VIII. Although the methods by which human genes are isolated today differ from the case described here, the example illustrates many of the general features of DNA cloning. Defects in the gene for factor VIII are the cause of the most common types of hemophilia, hemophilia A. This genetically determined disease has been recognized for over 1,000 years and affects approximately 1 in 10,000 males. People with hemophilia A fail to produce fully active factor VIII and thus have repeated episodes of uncontrolled bleeding. Until recently, the standard treatment for this disease was the injection of concentrated factor VIII protein, pooled from many blood samples. Tragically, before blood was routinely screened for HIV, the virus that causes AIDS, this treatment exposed many hemophiliacs to HIV infection. The commercial production of pure factor VIII using recombinant DNA technology thus offered a significant improvement in the treatment of hemophilia. This achievement required the cloning of the normal human gene that codes for factor VIII and the piecing together of its coding sequence. This DNA sequence was then used to produce large amounts of the purified protein, as we shall describe. Dealing with the 3 × 10⁹ nucleotide pairs of the complete human genome is a daunting task, and the first step in cloning any human gene is to break up the total genomic DNA into smaller, more manageable pieces. One general procedure for doing this is outlined in Figure 1011. Human DNA is first extracted from a tissue sample or cell culture and cleaved with a restriction nuclease, which produces millions of different DNA fragments. The mixture of DNA fragments is then ligated into plasmid vectors under conditions that favor the insertion of single DNA fragment into each plasmid molecule. These recombinant plasmids are mixed with a culture of E. coli at a concentration that ensures that no more than 1 plasmid molecule is taken up by each bacterium. The collection of cloned DNA fragments in the resulting bacterial culture is known as a DNA library. In this case, it is called a genomic library, as the DNA fragments are derived directly from the chromosomal DNA. If colonies derived from a single bacterium are isolated on petri dishes, each bacterial colony will represent a clone of one particular stretch of human DNA. A collection of several million colonies in this library should thus represent all of the human genome. To find a particular gene, we now face a problem analogous to that of entering a real library with millions of books and realizing that there is no card catalog or computerized listing to help us find the volume we want. How do we find a particular stretch of DNA, in our case, the factor VIII gene in the vast human DNA library? The key is to exploit the hybridization properties of nucleic acids discussed earlier in this chapter. If we had a DNA probe for the factor VIII gene, we could use it to find the matching clone in the library. But where did such a probe come from before the gene itself has been identified and cloned? In the case of factor VIII, a small amount of the protein was purified from human blood donors, using blood clotting as the biochemical assay. The partial amino acid sequence of the protein was determined. Today, this would be done using mass spectrometry. See Figure 445. Applying the genetic code in reverse, the amino acid sequence information was then used to deduce the partial nucleotide sequence of the gene. This nucleotide sequence was then chemically synthesized to create a DNA probe. Using this probe, the rare bacterial clones in the DNA library containing this complementary factor VIII fragment were identified by hybridization. Figure 10.12. When such a factor VIII probe was first used on a human genomic library, it picked out a single complementary clone. The nucleotide sequence of this clone DNA showed that it contained only a small portion of the factor VIII gene, and the entire gene had to be laboriously pieced together. We now know that the factor VIII gene is 180,000 nucleotide pairs long and contains many introns. See Figure 7.18b. So it is hardly surprising that no single genomic clone contained the entire gene. Many human genes were originally identified and cloned using variations on the procedure described for factor VIII. Now that the complete human genome sequence is known, however, cloning a particular gene is often much easier. For example, once the partial amino acid sequence of a protein of interest is known, it can, with the aid of computers, be used to directly search the human genome sequence for the gene that encodes it. Once the gene has been found in the electronic database, one can then design probes to pull the gene from a DNA library. It is even possible, as we describe shortly, to clone the gene directly from a sample of human DNA, bypassing the use of a library entirely. Cdna libraries represent the mRNA produced by a particular tissue. For many applications, it is advantageous to obtain a gene in a form that contains only the coding sequence, that is, a form that lacks the intron DNA. For example, in the case of the factor VIII gene, the complete genomic clone, introns and exons, is so large and unwieldy that it is necessary to work with the gene in pieces. Also, if one wanted to deduce the complete amino acid sequence of the factor VIII protein solely from the nucleotide sequence of its gene, it would be difficult to figure out where each exon begins and ends. After all, the great majority of the gene sequence is intron sequences. See Figure 7.18b. Fortunately, it is relatively simple to isolate a gene free of all its introns. For this purpose, a different type of DNA library, called a Cdna Library, is used. A human Cdna library is similar to a human genomic library in that it also contains numerous clones containing many different human DNA sequences. But it differs in one important respect. The DNA that goes into a Cdna library is not genomic DNA, but instead is DNA copied from the mRNAs present in a particular tissue or cell culture. To prepare a Cdna library, the total mRNA is extracted from the cells, and DNA copies of the mRNA molecules are produced by the enzyme reverse transcriptase. Figure 10.13. These complementary DNA, or cDNA molecules, are then cloned, just like the genomic DNA fragments described earlier, to produce the cDNA library. For example, using such a cDNA library prepared from liver, the organ that normally produces factor VIII, it was possible to isolate the complete coding sequence of the factor VIII gene, devoid of introns and present on one piece of DNA. The factor VIII cDNA was isolated from a cDNA library, using a portion of the genomic factor VIII DNA as a probe, by the procedure shown in Figure 10.12. We will see in the final part of this chapter how the coding sequence of the gene was used to produce purified human factor VIII protein on a commercial scale. There are several important differences between genomic DNA clones and cDNA clones as illustrated in Figure 10.14. Genomic clones represent a random sample of all of the DNA sequences found in an organism's genome and, with very rare exceptions, will contain the same sequences regardless of the cell type from which the DNA came. Also, genomic clones from eukaryotes contain large amounts of repetitive DNA sequences, introns, regulatory DNA, and spacer DNA. Sequences that code for proteins will make up only a few percent of the library. See Figure 929. By contrast, cDNA clones contain predominantly coding sequences, and only those for genes that have been transcribed into mRNA in the tissue from which the RNA came. As the cells of different tissues produce distinct sets of mRNA molecules, a different cDNA library will be obtained for each type of tissue. Patterns of gene expression change during development, so cDNA libraries will also reflect the genes expressed by cells at different stages in their development. By far, the most important advantage of cDNA clones is that they contain the uninterrupted coding sequence of the gene. Thus, if the aim of cloning the DNA is either to deduce the amino acid sequence of the protein from the DNA or to produce the protein in bulk by expressing the cloned gene in a bacterial or yeast cell, neither of which can remove introns from mammalian RNA transcripts, it is essential to start with cDNA. The main advantage of genomic clones, on the other hand, is that they contain introns as well as exons, and they include the regulatory sequences that determine when and where genes are expressed. For this reason, genomic clones are used to determine the complete nucleotide sequences of genomes, as we see later in this chapter. The polymerase chain reaction amplifies selected DNA sequences. Cloning via DNA libraries was once the only route to gene isolation, and it is still used in sequencing whole genomes and in dealing with very large genes. However, a method known as polymerase chain reaction, PCR, provides a much quicker and simpler alternative for many cloning applications, particularly for those organisms whose complete genome sequence is known. Today, the majority of cloning is done using PCR. Invented in the 1980s, PCR can be carried out entirely in vitro without the use of cells. Using this technique, a given nucleotide sequence can be selectively and rapidly replicated in large amounts from any DNA sample that contains it. For example, the polymerase chain reaction is now widely used to provide large amounts of any gene from a small sample of human DNA. It also has many other applications, including amplifying DNA for use in diagnostic tests for disease, genes, and in forensic medicine, as we discuss shortly. PCR is based on the use of DNA polymerase to copy a DNA template in repeated rounds of replication. The polymerase is guided to the sequence to be copied by short DNA primers that are added to the reaction and hybridized to the DNA template at the beginning and end of the desired DNA sequence. These primers provide the 3' ends for the DNA polymerase to start replication on each DNA strand. Primers must be designed by the experimenter and synthesized to order, so PCR can be used only to clone a DNA whose beginning and end sequences are known. During each round of replication, the 2 strands of the double-stranded DNA template are separated and copied independently. Figure 1015 shows the separate steps in the first round of replication. After many such rounds of replication, many copies of the sequence, typically billions, will have been made. Figure 1016. PCR is extremely sensitive. It can detect a single copy of a DNA sequence in a sample by amplifying it so much that it becomes detectable by, for example, staining after separation by gel electrophoresis. See Figure 10.3. There are several especially useful applications of PCR. First, PCR is now the method of choice for cloning relatively short DNA fragments, say under 10,000 nucleotide pairs from a cell. The original template for the reaction can be either DNA or RNA, so PCR can be used to obtain either a full genomic copy, complete with introns and exons, or a cDNA copy of the gene. Figure 1017. The beauty of this method is that genes can be cloned directly from any piece of DNA or RNA without the time and effort needed to first construct a DNA library. Another use for PCR, which relies on its extraordinary sensitivity, is the detection of infections by pathogens at very early stages. In this case, short sequences complementary to the pathogen's genome are used as primers, and following many cycles of amplification, the presence or absence of even a few copies of an invading genome in a sample of blood can be ascertained. Figure 1018. For many infections, PCR is the most sensitive method of detection. Already, it is replacing the use of antibodies against surface proteins to detect the presence of pathogens in human samples. Finally, PCR has great potential in forensic medicine. Its extreme sensitivity makes it possible to work with a very small sample, minute traces of blood and tissue that may contain the remnants of only a single cell, and still obtain a DNA fingerprint of the person from whom it came. The genome of each human, with the exception of identical twins, differs in DNA sequence from the genome of every other human. DNA amplified by PCR using a particular primer pair is therefore quite likely to differ in sequence from one individual to another. Using a carefully selected set of primers that cover known highly variable parts of the human genome, PCR can generate a distinctive DNA fingerprint for each individual. Figure 1019. Deciphering and exploiting genetic information. So far in this chapter, we have assumed that the function of a gene we may wish to study is understood, at least in rough outline. But suppose that you have discovered a gene that codes for a protein of unknown function. How do you determine what the protein does? Now that genome sequencing projects are rapidly identifying new genes from the DNA sequence alone, this has become a common question in cell biology. For example, of the 25,000 human genes that have been identified, see how we know, pages 318 to 319, more than 10,000 have functions that remain unknown. The procedures described so far enable biologists to obtain large amounts of DNA in a form that is easy to work with in the laboratory. Whether it is present as fragments stored in a DNA library or as a collection of PCR products nestled in the bottom of a test tube, this DNA provides the raw material for perhaps the most exciting experiments, those designed to unravel how individual genes and the RNA molecules and proteins they encode function inside living organisms. At this point in the process, biologists can be especially creative, because there are as many ways to approach the problem of gene function as there are scientists who wish to tackle it. The techniques that investigators use to explore the activity of a gene often depend on their background and training. Geneticists might choose to engineer mutant organisms in which the activity of the gene has been disrupted. Biochemists might use the gene to produce large amounts of protein to determine its enzymatic activities and three-dimensional structure. And those who are more computationally inclined might begin by searching computer databases for genes with similar sequences or expression patterns. Although these approaches differ in terms of the expertise and equipment needed to execute them, all are designed to yield clues about what a gene does inside a cell or organism. In this section, we present a few of the methods that are used to determine the function of a gene. Because all of these approaches begin with the nucleotide sequence of a gene, we begin by describing the techniques used to sequence a stretch of DNA. We then discuss how scientists can begin to interpret the information encoded in this nucleotide sequence. Next, we review how, once a gene is in hand, recombinant DNA technology can be used to produce enough of its RNA or protein product to conduct structural and functional studies. Finally, we describe several techniques for investigating what a gene does inside a cell, tissue, or even a whole plant or animal. These methods have revolutionized all aspects of cell biology by providing new ways to study the function of genes, RNA molecules, and proteins. DNA can be rapidly sequenced. In the late 1970s, researchers developed methods that allow the nucleotide sequence of any purified DNA fragment to be determined simply and quickly. These techniques have made it possible to determine the complete nucleotide sequence of hundreds of thousands of genes and the complete genome sequences of many organisms, including the budding yeast Saccharomyces cerevisiae, the nematode worm Caenorhabditis elegans, the fruit fly Drosophila melanogaster, the model plant Arabidopsis thaliana, and the dog, rat, chimpanzee, gorilla, and human. Several schemes for sequencing DNA have been developed, but the most widely used is Dideoxy Dna Sequencing, a method based on DNA synthesis carried out in vitro in the presence of special chain-terminating deoxyribonucleoside triphosphates. In this technique, DNA polymerase is used to make partial copies of the DNA fragment to be sequenced. These DNA replication reactions are performed under conditions that ensure that the new DNA strands terminate when a given nucleotide A, G, C, or T is reached. Figure 1020. These reactions ultimately produce a collection of different DNA copies that terminate at every position in the original DNA, and thus differ in length by a single nucleotide. These DNA copies can be separated on the basis of their length by gel electrophoresis, and the nucleotide sequence of the original DNA can be determined from the order of these DNA fragments in the gel. Figure 1021. Although the same basic method is still used today, many improvements have been made. DNA sequencing is now completely automated. Robotic devices mix the reagents and then load, run, and read the order of the nucleotide bases from the gel. This process is facilitated by the use of chain-terminating nucleotides that are each tagged with a different colored fluorescent dye. All 4 synthesis reactions can thus be performed in the same tube, and the products can be separated in a single lane on a gel. A detector positioned near the bottom of the gel reads and records the color of the fluorescent label on each band as it moves past, and a computer stores the sequence for subsequent analysis. Figure 1022. How researchers can piece together a complete genome sequence from this information is described in How We Know, pages 348 to 349. Of course, obtaining the sequence of a gene or an entire genome is only a beginning. As we discussed in a previous chapter, it is a great challenge to look at a nucleotide sequence and determine, for example, where a gene begins and ends, and which bits are important for regulating its activity. Even with this information, it is not necessarily apparent what role the gene plays in the physiology of an organism. To do that, biologists need to manipulate each gene more directly. Completely novel DNA molecules can be constructed. Studying gene function usually involves the construction of recombinant DNA molecules. As we have discussed, such recombinant molecules are generally made by using DNA ligase to join together two pieces of DNA, including DNA that comes from different organisms or DNA that has been chemically synthesized. See Figure 10.6. By repeated DNA cloning, DNAs from any source can be isolated and joined together in various combinations to produce DNA molecules of any desired sequence. Figure 10.23. The ability to generate custom-designed DNA molecules allows scientists to manipulate genes in a variety of useful ways. They can introduce mutations that alter the activity of a gene and approach that could reveal the normal function of the gene. They can also engineer hybrid proteins that contain, for example, a fluorescent marker protein attached to the protein of interest. Such protein fusions allow scientists to visualize where a protein is located within a cell or organism. Finally, in one of the most immediately practical uses of recombinant technology, scientists can direct the high-level production of what are normally rare cellular proteins, as we shall see next. Rare proteins can be made in large amounts using cloned DNA. Most of the thousands of different proteins in a eukaryotic cell, including many with crucially important functions, are present in very small amounts. For these proteins, it used to be extremely difficult, if not impossible, to obtain enough of them in pure form to be able to study them. One of the most important contributions of DNA cloning and genetic engineering to cell biology is that they make it possible to produce any protein, including the rare ones, in nearly unlimited amounts. This high-level production is usually accomplished by using specially designed vectors, known as expression vectors. Unlike the cloning vectors discussed earlier, these vectors also include appropriate transcription and translation signals, so that an inserted gene is expressed at very high levels. Figure 10.24. Different expression vectors are designed for use in bacterial, yeast, insect, or mammalian cells, each containing the appropriate regulatory sequences for transcription and translation in these cells. The expression vector is replicated at each round of cell division, giving rise to a cell culture able to synthesize very large amounts of the protein of interest. Because the protein encoded by the expression vector is typically produced inside the cell, it must be purified from the host cell proteins after cell lysis by chromatography. But because it is so plentiful in the cell lysate, often comprising 1 to 10% of the total cell protein, purification is usually easy to accomplish in only a few steps. This technology is now used to make large amounts of many medically useful proteins. The factor VIII protein, for example, is now made commercially from cultures of genetically engineered mammalian cells and is thus free of viral contamination. Many other useful proteins, including hormones such as insulin, growth factors, anti-cancer agents, and viral coat proteins for use in vaccines, are also produced in this fashion. The use of expression vectors also allows scientists to produce many proteins of biological interest in large enough amounts for the detailed structural and functional studies that were once impossible. With large quantities of a protein, scientists can analyze its biological or biochemical activity and perhaps even grow crystals that would be suitable for determining its three-dimensional structure by X-ray crystallography. See How We Know, pages 158 to 160. Recombinant DNA techniques allow scientists to begin with a protein of unknown function, isolate the gene that encodes it, and churn out enough of the protein to be able to investigate its structure and activity. Figure 1028. Reporter genes and in situ hybridization can reveal when and where a gene is expressed. Neither the complete nucleotide sequence of a gene nor even the three-dimensional structure of the protein is sufficient to deduce a protein's function unless it is closely related to a protein whose function is already known. Many proteins, such as those that have a structural role in the cell or normally form part of a large multi-enzyme complex, have no obvious biochemical activity on their own. Even those that do have known activities, protein kinases, for example, could in principle participate in any number of different pathways in the cell. In other words, it is not always clear from their biochemical activities how the proteins are actually used by the cell. In many cases, clues to a protein's function can be obtained by examining when and where its gene is expressed in the cell, or in the organism as a whole. Determining the pattern and timing of a gene's expression can be accomplished by joining the regulatory region of the gene under study to a reporter gene, one whose activity can be easily monitored. As we discussed in detail in Chapter 8, gene expression is controlled by regulatory DNA sequences, usually located upstream of the coding region, that are not transcribed themselves. These regulatory sequences, which control which cells will express a gene and under what conditions, can also be made to control the expression of a reporter gene. The level, timing, and cell specificity of reporter protein production will reflect the function of the original gene as well as the action of the regulatory sequences that belong to it. Figure 1029. In most cases, the expression of the reporter gene is monitored by tracking the fluorescence or enzymatic activity of its protein product. One of the most popular reporter proteins used today is green fluorescent protein, GFP, the molecule that gives luminescent jellyfish their greenish glow. In many cases, the GFP gene can simply be attached to one end of the gene that encodes a protein of interest. Often, the resulting GFP fusion protein behaves in the same way as the original protein, and its distribution in the cell or in the organism can be monitored simply by following its fluorescence by microscopy. Figure 1030. The GFP fusion protein strategy has become a standard way to determine the distribution and to track the movement of any protein of interest in a living organism. From this information, many insights about a protein's function in the organism can be obtained. It is also possible to directly observe the time and place that the mRNA product of a gene is expressed. In most cases, this strategy provides the same overall information as the reporter gene approach. In some situations, however, monitoring RNA is the method of choice. For example, if the gene's final product is RNA rather than protein. The technique, which relies on the principles of nucleic acid hybridization described earlier, is called in situ hybridization, from the Latin in situ, in place, because it allows specific nucleic acid sequences to be located while they are still in place within cells or within chromosomes. In situ hybridization uses nucleic acid probes labeled with either fluorescent dyes or radioactive isotopes to detect the presence of RNA or DNA of a particular sequence within a cell or tissue. Figure 1031. This technique can reveal gene expression patterns and has led to great advances in our understanding of embryonic development by making easily visible the many changes in gene expression that occur in different cells of the developing embryo. In situ hybridization also allows the visualization of specific DNA sequences within chromosomes. In this case, the probes are hybridized to follow chromosomes that have been exposed briefly to a very high pH to separate the two DNA strands. The chromosomal regions that bind the labeled probe can then be seen. Figure 1032. This technique can be used medically to identify early in pregnancy fetuses that carry abnormal chromosomes. Hybridization on DNA microarrays monitors the expression of thousands of genes at once. We saw in Chapter 8 that a cell expresses only a subset of the genes available in its genome. One of the most important uses of nucleic acid hybridization is to determine for a population of cells exactly which genes are actively being transcribed into mRNA and which are transcriptionally silent. In situ hybridization methods allow scientists to monitor the expression of one gene or relatively few genes at a time. In the 1990s, however, investigators developed a new tool called a Dna Microarray that allows the RNA products of tens of thousands of genes to be monitored at the same time. By examining the expression of so many genes simultaneously, we can begin to identify and study the complex gene expression patterns that underlie cellular physiology, visualizing which genes are switched on or off as cells grow, divide, or respond to hormones, toxins, or infection. DNA microarrays are little more than glass microscope slides studded with a large number of DNA fragments, each containing a nucleotide sequence that serves as a probe for a specific gene. The most dense arrays contain hundreds of thousands of these fragments in an area smaller than a postage stamp, allowing the expression patterns of entire genomes to be monitored in a single experiment. Some types of microarrays carry DNA fragments corresponding to entire genes that are spotted onto the slides by a robot. Other types contain short, single-stranded DNA molecules that are synthesized on the surface of the wafer with techniques similar to those that are used to etch circuits onto computer chips. In either case, the exact sequence and position of every DNA probe on the chip is known. To use a DNA microarray to monitor the expression of every gene in a cell simultaneously, mRNA from the cells being studied is extracted and converted to cDNA. See Figure 1013. The cDNA is then labeled with a fluorescent probe. The microarray is incubated with the labeled cDNA sample, and hybridization is allowed to occur. Figure 1033. The array is then washed to remove unbound molecules, and the positions to which fluorescently labeled DNA fragments have hybridized through complementary base pairing are identified by an automated microscope. The array positions are then matched to the particular genes whose DNA was originally spotted at each location. DNA microarrays have been used to examine everything from the changes in gene expression that make strawberries ripen to the genetic signatures of different types of human cancer cells. Comparisons of the gene expression profiles of human cancers, for example, can be used to readily distinguish one type of cancer cell from another. By relating these expression patterns to clinical data gathered for each cancer, including how rapidly it progresses and whether it responds to treatment, it may be possible to predict whether a particular patient will respond to a specific therapy. Thus, microarray-based profiles of cancer cells are likely to lead to much more precise and effective treatments for this often fatal disease. Genetic approaches can reveal the function of a gene. Ultimately, Cell biologists wish to determine how genes and the RNAs and proteins they encode function in an intact organism. Before the advent of gene cloning, the functions of most genes were discovered by identifying and studying mutant organisms. In this classical genetic approach, one begins by isolating mutants that have an interesting or unusual appearance, fruit flies with wide eyes or curly wings, for example. Working backward from this phenotype, the appearance or behavior of the individual, one then determines the organism's genotype, the form of the gene responsible for that characteristic. The classical genetic approach is most easily applicable to organisms that reproduce rapidly and can be easily mutated in the laboratory, such as bacteria, yeasts, nematode worms, and fruit flies. Recombinant DNA technology has made possible a different type of genetic approach. Instead of beginning with a randomly generated mutant and using it to identify a gene and its protein, one can start with a cloned gene and proceed to make mutations in it in vitro. Then, by reintroducing the altered gene back into the organism from which it originally came, one can produce a mutant organism in which the gene's function may be revealed. Using techniques to be discussed shortly, the coding sequence of a cloned gene can be altered to change the functional properties of the protein product or even to eliminate it altogether. Alternatively, the regulatory region of the gene can be altered so that the amount of protein made is altered, or so that the gene is expressed in a different cell type from normal or at a different time during development. The ability to manipulate DNA in vitro makes it possible to introduce precise mutations, and genes can thus be altered in very subtle ways. It is often desirable, for example, to change the protein that the gene encodes by just one or a few amino acids. The use of the technique of site-directed mutagenesis to achieve this is outlined in Figure 1034. By changing selected amino acids in this way, one can determine which parts of the polypeptide chain are crucial to such fundamental processes as protein folding, protein-ligand interactions, and enzymatic catalysis. Moreover, site-directed mutagenesis allows one to determine the biological roles of each part of a given protein. Animals can be genetically altered. To study the function of a gene that has been mutated in vitro, ideally, one would like to generate an organism in which the normal gene has been replaced by the altered one. In this way, the function of the mutant protein can be analyzed in the absence of the normal protein. In many organisms, such gene replacement can be accomplished quite easily by homologous recombination between the introduced mutant DNA and the chromosomal DNA. Figure 1035a. With the same strategy, the gene can be deleted entirely, creating a gene knockout. Figure 1035b. A third possibility is that the mutant gene can be added to the genome without any alteration being made to the normal gene in the process. Figure 1035c. Organisms into which a new gene has been introduced or those whose genomes have been altered in other ways using recombinant DNA techniques are known as transgenic organisms or genetically modified organisms, GMOs. In sexually reproducing organisms, these alterations are usually made in the germline, that is, in the reproductive cells. Such transgenic animals will then be able to pass the altered gene onto at least some of their progeny as a permanent part of their genome. Figure 1036. Technically, even the human germline could now be altered in this way, although this is unlawful for a variety of ethical reasons. Similar techniques are, however, being explored to correct genetic defects in human somatic cells. Somatic cells, such as the cells that form the liver, pancreas, bone, or skin, reproduce within an individual human being but are not passed on to progeny. See Figure 9.3. Some genetic diseases could be alleviated or even cured by the introduction of genetically corrected somatic cells into the tissue most affected by the disease. However, the alterations would not be passed on to descendants. Transgenic techniques make it possible to produce complex organisms that contain altered genes or that lack certain genes entirely. For example, many knockout mice, strains of mice that have a particular gene permanently inactivated, have now been produced. By observing the effects of eliminating or mutating a given gene, it is often possible to infer its normal function. Figure 1037. Rna interference provides a simple way to test gene function. Knocking out a gene in an organism and studying the consequences is perhaps the most powerful approach for understanding the function of a gene. But there is a much faster and easier way to inactivate genes in cells and organisms, called RNA interference, RNAi. This method exploits a natural mechanism used in a wide variety of plants and animals to regulate selected genes and to destroy foreign RNA molecules. See Figures 826 and 827. The technique relies on introducing into a cell or organism a double-stranded RNA molecule whose nucleotide sequence matches that of the gene to be inactivated. This double-stranded RNA is recognized as being foreign, and the cell is tricked into degrading not only it, but also the mRNA whose sequence it matches. Small fragments of these degraded RNAs are subsequently used by the cell to produce more double-stranded RNA that directs the continued elimination of the target mRNA. Because these short RNA fragments can be passed on to progeny cells, RNAi can cause heritable changes in gene expression. RNAi is frequently used to inactivate genes in mammalian cell culture lines. It has also been widely used to study gene function in the nematode C. elegans. When working with worms, introducing the double-stranded RNA is simple. The RNA can be injected directly into the intestine of the animal, or the worm can be fed with E. coli engineered to produce the RNA. Figure 1038. The RNA is distributed throughout the body of the worm, where it inhibits expression of the target gene in different tissue types. Because the entire genome of C. elegans has been sequenced, RNAi is being used to help in assigning functions to the entire complement of worm genes. Transgenic plants are important for both cell biology and agriculture. Although we tend to think of recombinant DNA research in terms of animal biology, these techniques have also had a profound impact on our study of plants. In fact, certain features of plants make them especially amenable to recombinant DNA methods. When a piece of plant tissue is cultured in a sterile medium containing nutrients and appropriate growth regulators, some of the cells are stimulated to proliferate indefinitely in a disorganized manner, producing a mass of relatively undifferentiated cells called a callus. If the nutrients and growth regulators are carefully manipulated, one can induce the formation of a shoot within the callus, and in many species, a whole new plant can be regenerated from such shoots. In a number of plants, including tobacco, petunia, carrot, potato, and Arabidopsis, a single cell from such a callus can be grown into a small clump of cells from which a whole plant can be regenerated. See Figure 829. Just as mutant mice can be derived by the genetic manipulation of embryonic stem cells in culture, So, transgenic plants can be created from plant cells transfected with DNA in culture. Figure 1039. The ability to produce transgenic plants has greatly accelerated progress in many areas of plant cell biology. It has played an important part, for example, in isolating receptors for growth regulators and in analyzing the mechanisms of morphogenesis and of gene expression in plants. These techniques have also opened up many new possibilities in agriculture that could benefit both the farmer and the consumer. They have made it possible, for example, to modify the ratio of lipid, starch, and protein in seeds, to impart pest and virus resistance to plants, and to create modified plants that tolerate extreme habitats, such as salt marshes or water-stressed soil. One variety of rice has been genetically engineered to produce beta-carotene, the precursor of vitamin A. If it replaced conventional rice, this golden rice, so called because of its faint yellow color, could help to alleviate severe vitamin A deficiency, which causes blindness in hundreds of thousands of children in the developing world each year. Essential concepts. Recombinant DNA technology has revolutionized the study of the cell, making it possible for researchers to pick out any gene at will from the thousands of genes in a cell and to determine the exact molecular structure of the gene. A crucial element in this technology is the ability to cut a large DNA molecule into a specific and reproducible set of DNA fragments by using restriction nucleases, each of which cuts the DNA double helix only at a particular nucleotide sequence. DNA fragments can be separated from one another on the basis of size by gel electrophoresis. Nucleic acid hybridization can detect any given DNA or RNA sequence in a mixture of nucleic acid fragments. This technique relies on the fact that a single strand of DNA or RNA will form a double helix only with another nucleic acid strand of the complementary nucleotide sequence. Single-stranded DNAs of known sequence and labeled with fluorescent dyes or radioisotopes are used as probes in hybridization reactions. Short DNA strands of any sequence can be made by chemical synthesis in the laboratory. DNA cloning techniques enable a DNA sequence to be selected from millions of other sequences and produced in unlimited amounts in pure form. DNA fragments can be joined together in vitro by using DNA ligase to form recombinant DNA molecules that are not found in nature. DNA fragments can be maintained and amplified by inserting them into a DNA molecule capable of replication, such as a plasmid. This recombinant DNA molecule is then introduced into a rapidly dividing host cell, usually a bacterium, so that the DNA is replicated at each cell division. A collection of cloned fragments of chromosomal DNA representing the complete genome of an organism is known as a genomic library. The library is often maintained as millions of clones of bacteria, each clone carrying a different DNA fragment. Cdna libraries contain cloned DNA copies of the total mRNA of a particular cell type or tissue. Unlike genomic DNA clones, cloned cDNAs contain predominantly protein-coding sequences. They lack introns, regulatory DNA sequences, and promoters. They are thus most suitable for use when the cloned gene is to be expressed to make a protein. The polymerase chain reaction, PCR, is a powerful form of DNA amplification that is carried out in vitro using a purified DNA polymerase. PCR requires prior knowledge of the sequence to be amplified, because two synthetic oligonucleotide primers must be synthesized that bracket the portion of DNA to be replicated. Historically, Genes were cloned using hybridization techniques to identify the plasmid carrying the desired sequence from a DNA library. Today, most genes are cloned using PCR to greatly amplify them and thereby obtain a specific sequence from a sample of DNA or mRNA. Techniques are now available for rapidly determining the nucleotide sequence of any piece of DNA. The complete nucleotide sequences of the genomes of hundreds of different organisms have been determined. These include bacteria, archaea, yeasts, insects, fish, plants, and mammals. Bacteria, yeasts, and mammalian cells can be engineered to synthesize large quantities of any protein from any organism, thus making it possible to study proteins that are otherwise rare or difficult to isolate. Using recombinant DNA techniques, a protein can be joined to a molecular tag, such as the green fluorescent protein, GFP, which allows the tracking of its movement inside the cell. In the case of GFP, the protein can be monitored over time in living organisms. In situ nucleic acid hybridization can be used to detect the precise location of genes in chromosomes, or RNAs in cells and tissues. By presenting a platform for performing a large number of simultaneous hybridization reactions, Dna Microarrays can be used to monitor the expression of tens of thousands of genes at once. Cloned genes can be permanently inserted into the genome of a cell or an organism by using recombinant DNA technology. Cloned DNA can be altered in vitro to create mutant genes that can then be reinserted into a cell or an organism to study gene function. A straightforward strategy for studying the function of a gene is to delete it from the organism's genome and then to study the effect of this knockout on the behavior or appearance of the organism. The expression of particular genes can be inhibited in cells or organisms by the technique of Rna Interference, RNAi, which prevents an mRNA from being translated into protein.